Welcome friends, my topic is structure and properties of hemoglobin. Hemoglobin is one of the commonest respiratory pigment and it is an iron containing and oxygen carrying red pigment. It is found in the erythrocyte of all vertebrate and in the blood plasma of some invertebrate like scarus, some flatworms, several annelids, arthropods and molluscs and sea cucumbers, etc. A modified type of hemoglobin called leg hemoglobin, which is found in the root nodules of the leguminous plant. And the normal hemoglobin found in the adult human beings is called hemoglobin A or HbA, and it is and that of the fetus is called a fetal hemoglobin or HbF. HbF has higher affinity for O2 than HbA has. It is synthesized in the fetus before birth. It may be almost completely replaced by HbA by the 12th month of after birth. Hemoglobin is a conjugated metalloprotein and it is formed of a colorless and globular basic protein called globin and an ion containing prosthetic group called hem or hematine. Hem is the active group taking part in O2 transport, and the hemoglobin varies with the species. Even in the same individual animal, there may be different kind of hemoglobin. All the different kind of hemoglobin have the same hem group but different globins, so they are different from each other only with regard to their globin part. This is the structure of hemoglobin A molecule. We can see alpha chain, beta chain, ion and hem group in this picture. A human hemoglobin molecule consists of one globin molecule and four hem molecule. The hem group is attached to the globin through a coordinate histidine nitrogen atom. The globin molecules in turn is a tetramer formed of four polypeptide chains, namely two alpha chains and two beta chains. In human hemoglobin A, and two alpha chains and two gamma chains in HbF. This is the main difference between HbF and HbA. These chains remain linked with each other and also within the hem molecule through weak non-covalent bonds such as van der Waals forces, hydrogen bonds, salt linkages, and hydrophobic interactions, etc. Each alpha chain is formed of 141 amino acids unit, and each beta, delta, or gamma chain is formed of 146 amino acids unit. The molecular weight of human hemoglobin is around 66,684 daltons. Hem molecule is an ion porphyrin complex. It is formed of an ion atom and a divalent porphyrin molecule. The porphyrin unit is a tetrapyrrole ring. It is formed of a 4-pyrrole ring with a centrally located ferrous atom. The porphyrin of hemoglobin bears a vinyl group on one of the 4-pyrrole ring. On the whole, it becomes clear that the hemoglobin A molecule consists of 574 amino acids, 4 polypeptide chains, 4 ion atoms, 4 porphyrin molecules, and a vinyl group. Hemoglobin has an active role in the transport of oxygen and carbon dioxide. Next is the properties of hemoglobin. The most important property of hemoglobin is its affinity for oxygen and its ability to reversibly bind with oxygen. A hemoglobin molecule can bind with as many as 4 oxygen molecules. Its affinity for oxygen depends on temperature, pH, and concentration of 2,3-diphosphoglycerate in the blood. The ability of hemoglobin to bind O2 enable it to serve as an oxygen carrier of blood. This reaction is unique in that the ion of Hem continues to remain in the ferrous state forming an oxyhemoglobin complex. The oxygen only loosely and reversibly combines with the ferrous ion without actually oxidizing it to ferric ion. So, the ion atoms get oxygenated and not oxidized. The oxidized ferric ion has no oxygen carrying capacity and so it is biologically inactive. The oxygen free hemoglobin is called deoxyhemoglobin. In adult human beings, there is yet another type of adult hemoglobin called hemoglobin A2 or HbA2. Its globin part is formed of two alpha chains and two delta chains. Each alpha chain is formed of 141 amino acids and each delta chain is formed of 146 amino acid residues. The hemoglobin content of the blood of a normal human adult consists of 97% of hemoglobin A, 2% of hemoglobin A2 and 1% of hemoglobin F. The normal level of hemoglobin A is in blood is 14 to 16 gram per dl in male and 13 to 15 gram in female. Thank you.